Hello and welcome to my presentation. I am Angela Saleh, a postdoctoral scholar at the University of Central Florida, working in Dr. King's laboratory. Our work consisted of a dose-dependent analysis of FDA-approved compounds for lubricin stimulation in a 3D human chondrocyte model of post-traumatic osteoarthritis, also known as PTOA. PTOA can start after a joint injury and represents about 12% of symptomatic osteoarthritis cases. Even with current care, we still do not have disease-modifying treatments for PTOA. Lubricin, also known as PRD4, a key protein for joint lubrication and joint balance, is often reduced. This can increase friction, cartilage wear, and synovial inflammation. So our approach was to repurpose FDA-approved drugs, because their safety is already known, to stimulate PRD4 in a translational 3D human chondrocyte model of PTOA. To screen more drugs at the same time, we adapted our 3D chondrocyte model from a 96 to a 384 well format. Primary human chondrocytes tagged with a PRD4 promoter Gaussian luciferase reporter were seeded in a U-bottom plate to form pellets. Pellets were fed every other day with basal chondrogenic media until maturation around day 12. Supernatant luciferase on day 12 or day 13 was used as the baseline, and I1 beta was added to simulate injury. After 24 hours, supernatant luciferase was measured to confirm the injury response, and the pellets were then treated with drugs or controls. Supernatant was collected every other day through day 21st or 22nd for luciferase measurement. For drug selections, we start with a library of 3,068 FDA-approved drugs, where we apply some exclusion criteria, such as removal of compounds that were not provided as 10 millimolar in the MSO, the same drug in different formulations, prodrugs, vitamins, and highly cytotoxic compounds. This gave us 1,273 eligible compounds, and 378 were randomly chosen for the first screen. For dosing, we estimated EC-IC9 from published EC-IC50 values. After the screen, the main PRD4 stimulators were tested in a dose-response study to find the dose that gives the highest PRD4 induction. To optimize the 384 well model, we changed key parameters for a 96 well plate, as shown in Table A. After these changes, we compared the 96 and 384 well models in a luciferase assay, using the TGF-beta1 as a positive control, because it's already known to have an effect in increasing lubricin expression. As we can see in B, this graph shows the normalized expression of PRD4 in y-axis and the days we measured in x-axis. The PRD4 induction was similar in both formats, with no significant difference. Here, in A, we have a schematic design of our reporter assay. When a drug increases a PRD4, it also increases the secretion of Gaussian luciferase, which we measure in the cell supernatant. In B, we have an example of a heat map profile from the luciferase screening with several tested drugs, which help us to visualize possible PRD4 stimulators. Overall, here in C, we identified 10 compounds that increased PRD4 expression on specific days compared with DMSO, that is our vehicle control. Using predicted target database for the 10 stimulators, we identify key genes and enriched functions linked to anti-PTO biology, including regulation of inflammation and connective tissue development. The dose response analysis revealed 3 top hits that we'll now be testing in a chondrocyte from patients of different ages and sexes, and as well in a PTOA mouse model. So as conclusion, the 384 well format was optimized. From the 378 FDA screened compounds, 10 were identified as PRD4 stimulators and several hits produced PRD4 reported activity comparable to or exceeding TGF-beta 1 at selected time points. Hit mechanisms or targets cluster into PTOA-relevant biology supporting target-based selection for follow-up validation. And the top 3 hits showed consistent dose-response curves within nanomolar ranges. We'd like to thank you all, those involved in this work, and you as well for watching my presentation.